search patterns
This
for
meeting is
life
being recorded.
Oops. From this idea of thinking not just about space, but about time as well. And I want to start out by inviting you all out to venture out into the solar system with me. And I'd like to start with this image that we see here. This is this vast sea of stars that we're all sitting in right now in front of our computers in a terrible storm. And if you look at this picture, you'll see there's tiny clumps in this image. Some of those are planets, and those are clustered around stars. And this is a vast sea of what you might even consider unknown knowns. There's all sorts of possibility space around us in the universe in the sea of stars. And there's many things that we can know about that, though, since we know that the physical laws that direct matter and motion are just as valid out there in the solar system as they are here. So we know how these laws direct chemistry and geology, atmosphere building, distributions of elements. These are our knowns. These are the physical laws that we look out into space and we understand. But the specific predictions of conditions at these stars are often really hard, since we're really thinking about any particular state that needs to have the information of its starting material. And this is a theme that we'll return to over and over again tonight. And that starting material has this cascade of branching possibilities that's open to it at any point. And that starting material for many of the ways that we're going to think about it tonight are the planets. So in this expanse of possibilities, the pursuit that I'm particularly interested in science, and that I'm hoping to engage all of you in tonight, is the search for something very special, and that something is life. I wanted to point out this tiny dot that's on your screen right now. And that is the only place where we know there is life. That tiny dot is your planet and my planet, Earth. This is an image that was taken by the Voyager spacecraft as it sped past Neptune. It turned around one last time to take this family portrait of all of us. And so in this vast sea of possibility space where we're thinking about life, this is the one place where we can really do those experiments to try and understand what is the nature of this thing that we call life. So everyone you've ever known and ever loved, all the life that encapsulates our planet is right there in that tiny dot. And I hope that gives us all a little bit of perspective for what it's like to look for life in space. Now, I know that many of you are astronomers or interested in astronomy, and this is a little bit different talk than you might always have. So I'm going to start out by trying to frame the search for life as we have it, and think about how do we look for life. And of course, when we think about looking for life, for those of us that are living, everyone on this Zoom call, we have an intuition for what we think it would look like. And that's because, of course, we are living on this life-saturated planet. We're surrounded by life. We have accumulated observations that we've all made over the course of our lives, and we've made assumptions about what we think life is based on that rich experience of our own time here on this planet. So we have an idea based on these observations of what the necessary features are when we go looking for life someplace else. And we can apply these ideas of what we think life are at scale. So the microbe is following many of the same sorts of biological imperatives that the elephant is. And in fact, it even shares a huge amount of its biochemistry with the elephant, even though it's a very different scale. So these things that we think about life span scales. And what we've learned from the elephant, we can apply to the microbe. Those traits and processes, even though they're at drastically different scales and different lifestyles, are just as valid. But how does that help us when we're thinking about finding life elsewhere? And this is where the idea of time machines comes in. Because a lot of the way that we think about life and astrobiology and that search is something that comes to us from paleobiology, which is actually kind of the core discipline that I was brought up in through my graduate work. And this makes sense in many respects because the past really is another country. If I suddenly transported everyone on this Zoom call back to 6 million years ago on the beaches, yes, the beaches of Alberta, That's how different the world was back then. We would see the teeming creatures on the seaside that left behind this fossil imprint we see here. And these organisms are wildly different than anything that we see now. We don't have a living predecessor of these organisms. And so looking at ancient life helps us open up our creative space about thinking about life elsewhere in the universe by challenging all of those same assumptions. How is this the same or different than the elephant or microbe, given that it was a very different organism, and even the world it lived in is very different, even though it's the same world we all still share. So similarly, when we see organisms, we have to think about what they tell us about antiquity. What I'm showing you here, many of you may think that this is rocks on a beach, but all of those rocks that you see there are actually what we call stromatolites. And stromatolites are webs, not webs, they're kind of gooey masses of microbes that build up and collect sediment. So they actually kind of build their own rocks and homes, and they grow in layer upon layer over time out of the seabed that you see them there. We could think of them as the earliest example of skyscrapers. That's microbes building layer upon layer, accumulating sediment from the sea around them. And this is a kind of lifestyle in an organism that we find all throughout the fossil record. Their fossil record stretches back 3.5 billion years to the very dawn of when we start to see evidence of organisms in the fossil record. So these stromatolites give us a really strong way of using time machines and thinking about the past to tell us about what we can look for when we're thinking about life, the conditions and features that will tell us about life. But the fossil record also gives to us this cautionary tale about trying to replay time too prescriptively. We can overinterpret the fossil record and assume a bunch of parsimony and try to predict what organisms might look like. But organisms are an accretion of adaptations that were forced upon them by the environment around them. Life records adaptation of its environment and what that organism can be, or its totality in the past is unrecognizable without that context of deep time. I always like to give this example of the Riney Chert, these 400 million year old rocks from Scotland. There's these tiny little 3-centimeter things that you see there, and those are actually the earliest land plants. That's their stems. And if we were to go back to that seaside in Scotland 400 million years ago and look at those tiny little organisms living on the shore, we probably wouldn't predict vast forests in the Amazon or something like that, because what has become the vast forests of the Amazon is this accretion of adaptations unrecognizable without that context of deep time throughout just Earth's history. So what we need to think about when we're imagining life in the outer solar system or close to us in the solar system or in other galaxies around us is, how do we explore beyond our common heritage? Every example that we have of life on this planet shares biochemistry and shares heritage, but that doesn't mean that organisms everywhere would share the same heritage as us. We have to think about ways to imagine other origins of life. And so we have to think more generally about what we would expect from forms and potential biochemistries that could be around other planets. So paleobiology gives us these tools for understanding life in these alien contexts. So an example that I try to give when we're trying to imagine a truly alien environment is actually something very close to home, and that's the moon Titan. Titan is covered in hydrocarbon seas. So imagine seas of gasoline instead of seas of water. And it's very, very cold, and there's these icebergs that float on these seas. And so this is an utterly alien landscape. But there are things that we can learn from paleobiology that can tell us about biological potential, even at this planet. But we wouldn't be able to use any of the examples of life on this Earth for that search because it's so wildly different. So yes, how can we search for life without presupposing these particular forms or chemistries? When Sean was introducing me, he talked about the the lab group that I am a head of, and that we're focused on agnostic biosignatures. And this is what we mean when we say agnostic biosignatures, is biosignatures, signs of life that are agnostic to the origin or heritage of that life. We're not presupposing that life in the solar system would be related to us. We're trying to divorce ourselves from those assumptions. This, of course, brings us to the question, what even is life? And this is kind of an esoteric place for us to start. And of course, there really isn't a great working definition for life. Those of you who are in the physical sciences might be upset to know there's not really a strong definition. But what we try to do, rather than defining it, is thinking about what life is and does that can help us recognize the presence of life elsewhere. And this is actually a pursuit that people have been thinking about for quite some time. I like to mention this book. It's sitting on my desk back in my office. This was written by Erwin Schrödinger, the cat guy, the physicist. And he was very interested in this idea of what is life. And just like we do in my lab group, he tried to apply physics to it as a way to try and define the actions of life as a way of understanding what it is. And that's very much what we do in our group as well. And one of the fundamental ideas that comes to us from Schrödinger that helps us frame the search for life in an agnostic way is to remember that life, primarily, whether it's an elephant or a microbe or a stromatolite, life is an energetic response to an environment. I like to give the example of the Newton's cradle here because that's something that's familiar to us from physics and tells us a lot about physical environments. If we think of the planet or the galaxy as that Newton's cradle, where there's a lot of expected activity, be that chemistry, be that physics on a planet, there's expectations that we have about how nature works that comes to us from physics. And life does something a little bit different. Life is harnessing energy to do something that defies the rest of physics that is acting on the environment, that entropic force. So you see that one ball in the Newton cradle is going up a little bit farther than we would have expected if we were just rocking those balls that are swinging back and forth. That is what life does on an environment. And so that's how we can recognize it when we're looking for life in the solar system, is by that input of energy into a system that makes it look different. I'm going to give you some examples of how we think about life in this general framework that comes to us from Schrödinger and others that helps us understand how to look for life. This is also a very unsatisfying definition of life, but one way to describe you and me and every other organism around is that we are open systems with fluxes of matter and energy that are far from equilibrium thermodynamics. Going back to the Newton's cradle, that little ball that was swinging up, it was far from equilibrium thermodynamics. And so are you, and so am I, and so are all the organisms we share this planet with. You can think of it in the sense of elements that our corporeal forms as organisms are taking in and moving around matter and energy from our environment in a way that is out of equilibrium with the planet itself. And I'll show you some examples of that in a minute. Another way that we could describe life in these very general ways is to think of what is the nature of life when we think about that use of energy, metabolism. This is something that Schrödinger and others like to describe as the use of free energy to maintain structure. We're using energy, we're using matter from the planet around us, and we're maintaining structure in a way that is otherwise unpredictable or not predicted by the physics of a planet. So organisms arrange matter and energy into highly ordered structures with intrinsic complexity. And this is what makes us differ from the stars, is that intrinsic complexity. And we'll talk more about that in a minute too. I like to give this example of the way that intrinsic complexity shows up in nature at a lot of different scales, not just between organisms of different scales, but within an organism at different scales. So leaves are something with a lot of intrinsic complexity that are very different than what you might otherwise expect carbon to accumulate as on a planet's surface. They take these great shapes that are responsive to their environment, that have a lot of different jobs for that organism. And then if you look at them even closer, you see evidence of the cells and how there's communication with the cells that is driving this magical thing we call metabolism that is helping it maintain that structure. Within those cells, we see those green balls. Those are full of an energy harnessing molecule that is doing a bunch of jobs for that organism by providing elements for the continuation, bringing in that matter, and also generating energy. So this is brings in those fluxes, the relationship between organisms, energy, and matter, and it also shows us that intrinsic complexity that is being maintained through that use of free energy. So how does that help us understand what's going on out in the solar system? I know that's what everybody came here, is to think about space. Now, a lot of what I do and other folks here at NASA are doing is looking for biosignatures. We're using those very general ideas about life, many of us. Some people are looking for life that might have common heritage with Earth, but many of us are thinking about it in this general sense. And we use tools like rovers on other planets or telescopes, and we look out there and we look for what we call biosignatures. And that's a little bit of jargon. I promised Sean that I was going to try to minimize the amount of jargon, graphs, and organic chemistry, and you guys can keep score here. But I will define this little bit of jargon that I have. When I say we're looking for biosignatures, whether it's on a rover or on a telescope, what I'm really saying is that we're looking for an indication of past or present life. Does that mean I'm looking for an elephant? Probably not. Does that mean I'm looking for a microbe? Maybe. But what it really means is that we're looking for something that shows us evidence of matter and energy harnessed for structure. The elephant and the microbe are both examples of matter and energy harnessed for structure. And back to our paleobiology example, so are all these fossils and bones that you see on your screen. When you hear people talk about biosignatures in terms of the search for life in the universe, you might think that's something very complicated, but many of these things are very intuitive, like a fossil. They can be very complicated. Another really powerful biosignature that's out there is DNA. And of course, we're all familiar with how DNA is used to understand the presence of organisms. It's television's favorite molecule. And of course, this is part of that shared heritage, though, but it represents a complicated molecule with complexity that's intrinsic to it. So we understand that matter and energy were harnessed to build that structure. Footprints could be another great example of a biosignature, though. It doesn't necessarily have to be something about that organism. It can be an imprint the organism left in the environment, just like the fossil. So this is an example of lots of biosignatures that have been left by astronauts on the moon. That we harnessed a lot of matter and energy for structures to go there and leave behind these biosignatures. I always like to give the most complicated example that we're not going to talk about too much during the rest of our time together, but a great idea about biosignatures is really looking for energy itself. That is a way you could look for life. You could look for motion. And the example that I'm giving here is a way that we look for life on the sea floor of our own planet. You might imagine that we just go down there and collect DNA or look at fish, but this is a device that has been built by my colleague Pete at Harvard, and they use it to actually measure electrons moving across a sediment gradient. That's part of an extracellular electron transport mechanism that these organisms have. That's that cartoon that I show there. And this is a great way to illustrate what the Nobel Prize-winning biologist Albert Szent-Györgyi said, which is life is nothing but an electron looking for a place to rest. That is a very complicated way you could look for life, but it is a really great example of looking for energy itself and that relationship that life has with energy. So we're going to break this down into a couple of really brief sort of ways of taking all that information that helps us not define life, but describe life in physics sorts of terms and think about what that really means as a search pattern, because that's what we came here to talk about, is search patterns for life. And one really important search pattern that we've basically kind of walked ourselves into through those past examples is the redistribution of materials that represents energy. So you don't necessarily have to go looking for an electron. You can look for the redistribution of materials that doesn't follow the laws of physics, and understand then that there's the potential for past or present life that brought you that accumulation of materials. So here's some examples of how we would see that in hand sample or just on a landscape. Here are some examples that show us that there's patterns that arise in rocks when there are organisms that are part of the accretion of those sediments. So what you see on the left there may look not like an organism, because what you see on the right, that's clearly a trilobite. It's not a very big one. Trilobites have a long and glorious fossil record. Some of them are big, some of them are small. This is kind of a small one. And you might not look at the one on the left and think that looks like a biosignature. But what you find if you hang out with a lot of experimental sedimentologists is that when sediments are moving around in waters without the presence of all the sugars and goo and lipids that we associate with life, sediments will actually arrange themselves differently. And so you can do flume experiments in your local geology department and look at how sediments actually arrange themselves differently. It's a very subtle difference, but it is a difference. And this is something we think about when we look at ripples, kind of like what you see on the left, on Mars, and we try to understand if we think that there's the presence of any sort of organics that may have represented some sort of presence of life. I don't know if that example on the left necessarily has that. I'm not that kind of sedimentologist, but that is a way that you can represent a biosignature. Here's one, though, that we do know represents life, and that is what we would call a paleomat. That stuff that you see around that lens cap is a pattern that's left on a rock when there is a layer of biological material, a microbial mat that then dried up. This is very, very old. These are, I think these rocks are on the order of 400 or 500 million years old. But that was a mat that was on that beach all those years ago and has left behind this pattern that is unlikely, probabilistically unlikely, through the laws of physics that affect rocks. So this is a way of us looking at the biological processes that leave behind fabrics and sediments. I also like to show these examples because on the right, of course, here's a very familiar example to us of the way biology would interact with a sediment and leave something like a tire track. What you're seeing on the left is a little bit more, even more subtle example of redistribution of materials that represents energy. If you look at that pile of rubble, it probably looks like a lot of rocks, but if you have a trained eye, you can look at that and see that there's actually pieces of pottery there. This is pottery shards from a site of archaeological interest in New Mexico where I do field work. And I'm not an archaeologist, but you know, we meet each other in the field, and these are very strong biosignatures of human culture in that area in the relatively geologically recent past. And this is what could also be considered a redistribution of material that represented human energy and left this behind. So the other thing that's important to think about with this redistribution of materials is that it not only represents energy, but it represents a selection mechanism. Because remember, we were saying that all of these adaptations that bring us the organisms that we have now have come through history in this long conversation with their environment and landscape that proffered an advantage to certain expressions over others. So when we're seeing that redistribution of materials as an organism, we recognize that there was some impetus for that redistribution to appear the way it does, whether it's something about physical structure or chemical structure. There was a selection mechanism that brought us that. And that's what makes all of these sorts of expressions very different than what we would see out in space or on a planet without life. We'll talk a little bit more about that too towards the end. So a way of thinking about that, we'll go back to this example of fluxes of matter and energy. And here we can describe life as this force that concentrates scarce elements into discrete metastable entities that are geochemically distinct. This is probably my third rather unsatisfying way of describing life. For those of you that are keeping count of this, but you, me, and all the organisms we share this planet with are discrete metastable entities that are creating a geochemically distinct structure from relatively scarce elements. One way that you could think about this is that you and I are unlikely blobs of carbon on an otherwise silicate background of this planet. And that is the metastable, geochemically distinct structure that we're bringing to the surface of this planet. So since I was talking about carbon, I thought I'd show you some examples of how we can look at this with a kind of microscopy that helps us understand the presence of certain elements. Since we're talking specifically about elements, we talked about structure. Now we're going down a level in chemistry to thinking about elements. This image that you're seeing here is a microbe taken from the sea, where the red is showing us high concentrations of carbon bonded with a certain kind of nitrogen. This is a very common bond in organisms. And so you can see that that organism has much more of that type of carbon connected to a nitrogen than the waters around it. That is one way of us recognizing that this is an organism without knowing what taxa it is, without sequencing its DNA, without a particularly strong microscope. This is the kind of technology that we call heritage. This is the kind of instrument that has flown on many missions. We can look at elemental abundances all around our solar system. And so this is a way that we could look for life. It doesn't just have to be carbon, because remember, we're trying to not make assumptions about biochemistry based on our own. We could look at other element systems too. And so here, the same microbe, you can see that it has all these anomalous accumulations of phosphorus in it that you don't see in the waters around it. And even other elements that we don't necessarily always consider to be part of our own biology, things like copper, those are probably parts of enzymes. Metals are often parts of enzymes. And even though there is diffuse copper in the water around it, you're still seeing that metastable, geochemically distinct collection of copper in that organism. So here's some more compelling or charismatic examples of that. You are a carbon blob on a silicate background. This fossil leaf from 65 million years ago is also an anomalous bunch of carbon in a silicate background. So we can look around ourselves and see the way carbon is expressed now as part of organisms, and we can go back in time in our in our time machines and look at in examples from life's history to see how carbon was arranging in as part of our heritage. Here's another example of anomalous accumulations of geochemically distinct metastable entities. Both the fish and the tire fit that definition. Again, emphasizing the fact that biosignatures aren't always about the organism itself, but an effect it had on the environment, be it through something it made or something it left behind as an imprint on its environment. The tire is just as much a biosignature of human activity as bones we might leave behind. And of course, the fish is a very strong indication of life. I like to show this sort of example of me working in the field with my colleague, Christine. Here's where we're looking for life, kind of using these same sorts of tools. We're using analog tools that are aboard rovers that are on the surface of Mars right now. And we're testing out these kinds of biosignatures by just looking at elements and minerals. And we're looking in very unlikely places like the insides of lava tubes. This is a field site that is, as of December year before last, it went below lava, so we'll never be able to return here. This is on the flanks of Mauna Loa, but we're using those field scale instruments like around rovers, and we're looking for life just by looking for these maps of elements and minerals that can tell us that life either has had an effect on that rock or has not. since there are places on this planet where life is much more scarce and maybe hasn't had an effect that is necessarily visible. So we call these elemental maps and energy maps. And this is also getting us to the idea that not only is redistribution of materials something that represents inputs of energy, but all of that requires energy. And we won't talk too much about metabolisms, but when we look at minerals and we look at available elements, we can look and say, oh, this is a place that would be a much more likely habitat because there is all of this free energy that an organism could use to maintain that structure, to move those materials around, to obey those selection mechanisms that are making some things more adaptive and more optimized for that environment than other things. We can actually look habitat first and try to think about if something has interacted with it and left behind mineralogical evidence of life, or if it might become a good habitat at some point in the future of this rock. We've looked at it at field scale in a cave. Now here's another way that you can think of that redistribution of materials that life is doing at planetary scale. We, of course, know that we can try and look for biosignatures in atmospheres of exoplanets, but this is a really strong biosignature that we can see even on our own planet at huge scale. That bar that you see there in the corner is 50 kilometers. And what you're seeing here is a bloom of plankton. This is, I think, off the coast of Maine. This is such a vibrant activity that happens every year that we can see it from space. We can see those swirls, those white swirls. That turbidity in the water is the presence of life. And so if you were to see this sort of activity in another planet and you don't have a sedimentological reason to think that that's something coming from the shore, that would be a very strong biosignature because this is a really powerful redistribution of materials. This is a powerful redistribution of carbon into cells in that water that we can see from very, very far away. So I wanted to return for a minute to the example of the leaf. And I want to return to this because the leaf is telling us a couple of different things that help us understand this general framework that we have for life. And it also helps me explain a little bit about chemistry. I promise I'm not going to do a whole lot of organic chemistry, but it is my home discipline. And so I'm very weak and I might slip into it. But the leaf is a way that we can think about physical structure. Kind of like we were thinking about fabrics on rocks, kind of like how we are thinking of fossils that might be part of rocks that represent that accumulation of carbon. We can look at that structure and say that's intrinsically complex. It has selected reasons that it is that shape, and it took a lot of energy and matter to make that shape and maintain that structure. And then we look at it at closer details and we see that intrinsic complexity is happening at various levels. Leaf, cell, the chloroplasts that are full of chlorophyll, and intrinsic complexity then even extends to when we look at organic or chemical molecules themselves. Here's an example of an intrinsically complex molecule. This is chlorophyll. And you can see, for those of you who aren't familiar with structures of molecules, when we see this, when you see these bars, you can imagine that every vertex of the bar is a carbon. And so what this is showing us is the shape, this very unique shape of carbons. And this is not a shape that is normally created by a whole bunch of carbons hanging out together in space or someplace else. Remember, carbon comes from stars. It's all throughout our solar system. I'll show you some measurements of how we look at carbon throughout the solar system. Carbon is part of the elements that come to us from stars. But when we look at the kind of carbon chemistry, organic chemistry that's happening in space, we're not seeing these kinds of structures arising in abiotic settings where there isn't a lot of energy going into it to make these kinds of really unique structures. You also see a bunch of oxygens there. That's the red O's. You see nitrogens. That's the blue N's that you see. And you even see a magnesium element that is in the center of that molecule right there. So this molecule is telling us something about intrinsic complexity. Now, in my group, since we are mostly organic chemists, this kind of intrinsic complexity is very much a part of biosignature search patterns that we employ on other planets. This is the kind of data we're looking for. We're looking for these kinds of structures when we look at organic chemistry that might be on the surface of Mars or in the oceans of Europa. We're trying to find this kind of intrinsic complexity. I'm showing some molecules here that are all things that we would think of as being highly structured molecules with a lot of intrinsic complexity. These are all molecules that are part of organisms. Cholesterol is a big part of making cell membranes. The one in the middle is the energy molecule that's inside all of the organisms that we share heritage with here on Earth. Of course, chlorophyll. Now we've seen a drawing of, here's what its structure would look like on the right. Again, all of those black balls at the vertices would be carbon. These are very unlikely structures for carbon to take without a whole lot of energy and a selection mechanism driving the production of these particular molecules. And one of the things that we do in my group is we ask the question that without making assumptions about structure, is there a threshold above which molecules are unlikely to arise without biology? That machinery that's directed by selection that puts all of that effort into building these very particular things. I show one example here of a molecule that we do associate with life. That's glycine. It's an amino acid, but it's important to note that this amino acid and pretty much all of the amino acids that are part of life are also found in extraterrestrial settings. Not from organisms, but because amino acids are made as part of alteration processes from carbon-bearing material in asteroids. And we'll talk a little bit about OSIRIS-Rex towards the end of this. So a lot of these molecules that we often associate with life are actually part of astrochemical processes as well. So it becomes very important for us to employ all of these things we know about life and the relationship between life and energy in a directed way that's cognizant of the abiotic background, since there's all sorts of false positives out there because we share a lot of chemistry with our surrounding universe. So what we do in the studies that we do in our lab is we think about the simplest way to construct a molecule from its parts, including the simplifying feature of duplication. This is something that we've taken not from chemistry, but from graph theory, where we think about each of these sorts of parts of a molecule as basically the same way you would in graph theory, where it's vertices and nodes. And so we count up the number of steps, and we have programs that do this for us. And they say how many independent bonding steps each of these molecules would need in order to be made. And so here's an example of when we try to construct certain molecules from databases with this algorithm, and we look at the number of steps it takes. And so basically, if you look at some of those smaller molecules that are lower molecular weight, like asparagine and tryptophan, those are amino acids that we all have as part of our biochemistry, but are also being made in space. And you can see those have fairly small molecular assembly index numbers. That molecular assembly index number is the predicted number of least steps that any system would take. If it's following physics, this is the number of steps it would take to make that molecule. And you're seeing numbers like 7 and 12. But then when we look at other molecules that are only associated with biology, like penicillin or ATP, we see very high numbers of steps. And so that number of steps tells us that an organism put a bunch of energy into making enough duplicates of this molecule that it must be important, that must be that selection mechanism. And it's doing it in a way that's probabilistically unlikely in abiotic systems outside of biology. So once you get above maybe 15 or 16 steps, it starts to become very unlikely that you ever see it in an abiotic system. And I swear this is the last organic chemistry. I won't torture any of you anymore with organic chemistry. But I did want to return to this idea of chemistry and how powerful it is for when we're looking for biosignatures. When we talk about rovers or telescopes, there's two big things that we're looking at. We're looking at basically pictures and chemistry. That's pretty much all of biosignatures, because we don't have great ways of looking for electrons like my colleague does at the sea floor. This is a lot of what we're doing is cameras and chemicals. And so understanding this relationship between fabrics and sediments and the way we can look at objects and say how likely they are is, of course, the camera end of things. And then when we're thinking about chemistry and elements and minerals, that's the chemistry end of things that we're doing both with rovers and orbiters, be it Mars, be it Europa, or with telescopes like we use for looking at atmospheres of exoplanets. And both of these things require a bunch of knowledge and predictions about what you are going to see in abiotic chemistry, what you're going to see in chemistry that would naturally be happening in the energetic systems of the stars and planets themselves. So we require this prior knowledge of an expected abiotic chemical expression given the rules of a physical system. This is, of course, why I, as a biosignature person, am also part of the astrochemistry laboratory here at Goddard, because I'm trying to be a part of this observation of the solar system that's helping us understand what would arise outside of biological systems. So when we see that perturbation, when we see that ball that's going a little bit higher in the Newton's cradle, that's life, inside a chemical system, it's easier to recognize that that's a probabilistically unlikely event that could be life. So here's some of the data that we look at when we look out into stars. We see all of this rich chemistry happening. So when we talk about chemical complexity, we can't just say, is it a big molecule, or is there lots of them? Is there lots of chemicals? That's not the kind of information that we're thinking about. We're thinking about actual abundances, high abundances of molecules that would have a lot of steps that go in to make them. All of these molecules that you see here with this infrared telescope, the Herschel instruments, these are all fairly small. There's a lot of them, about 100 when they look out into interstellar media like they're doing right here, but they're all fairly small. And there's not actual patterns of a lot of selection for particular molecules like you and I have. Here's another example of looking at very, very large molecules. Remember I said, you know, those are small. Well, here's a way of looking at very, very large molecules. These are what we call PAHs, polycyclic aromatic hydrocarbons. They're massive. They're lots and lots of carbons. But they're not showing us any sort of selection mechanism. They're just basically the same, what we call chemical motif over and over and over again. So they don't have a whole lot of those steps that tell us that there was energy that went into making this. But it's important for us to remember that you and I record this inheritance from the stars as part of our biology. So much of what we have harnessed as organisms on this planet reflects the abundance of molecules that came to us from meteorites and were part of our planet from the very beginning. The fact that you and I have all these amino acids that we've harnessed to do metabolic work for us is very much a reflection of the fact that this is a lot of what has come to our planet from chemistry happening all around us in the solar system. We are part of this, and that pithy idea that you're made of stardust is actually really true. And this is a way of us thinking about how we differ then from the abiotic context that surrounds us. This, of course, brings us to the OSIRIS-Rex mission. This is a mission that's near and dear to my heart. When I'm not in this room talking to you guys, I'm going to be down the hall analyzing samples that I just got a couple of weeks ago. This is a spacecraft that went to a near asteroid, the asteroid Bennu. And what you're seeing here on your screen is this kind of Roomba-like device that we landed on there just for a few seconds and grabbed up a whole bunch of rocks from a little bit below the subsurface of that asteroid and brought them back home. When we think about sample return, we often think of the lunar return, and we do have many moon rocks, but sample return is also something that we do a lot with these sort of airless bodies. The Hayabusa mission to Ryugu, our mission to Bennu, we've captured comet particles and solar particles. And this is a very new sample that's helping us understand more about what that organic inheritance of molecules that comes from the stars is, and helping us also build that time machine to help us understand the starting materials of biology itself. I promised that I'd only have one graph, and here it is. This is a graph showing us the immense diversity of molecules that is inside meteorites that are part of our planet, and of course, interrupting and colliding with our planet all the time. What this piece of data shows you is every one of those dots on this graph is a different chemical formula. This is from a meteorite, the most famous meteorite, the Murchison meteorite. And what you're seeing here is nearly 10,300 individual molecular formula identified in this rock. So this tells you about the diversity of materials out there. And we as organisms select and only take just a few, and then we build up our own so that we have a very narrow bunch of chemistry that we're using as part of our organisms, whereas there's a vast chemistry surrounding us in space. Here's just some of those molecules, many that you would associate with us as organisms that are inside meteorites. Some of them are very large and don't dissolve in solvents, which makes them rather inaccessible for organisms such as ourselves, which require solvents. But many of these molecules are, of course, things that are part of our current biochemistry and that have been a part of our planet since our origin as living beings. So why do we study Bennu? It really is a way to take us back in a time machine to understand the earliest Earth. Those extraterrestrials would have been a major source of prebiotic organic ingredients that would have been part of that earliest biochemistry. And it's not just about the past. These sorts of materials and organic molecules are raining down on us all the time. We probably collect about 30,000 tons of cosmic dust here on the Earth in the form of micrometeorites and interplanetary dust. You've probably seen those demonstrations of how it's probably in your gutters of your house. Well, not with this rainstorm, it's all washed away. Of course, even more of that material was landing on the planet during the heavy bombardment period, just before the origin of life on our planet. So that's why we go to Bennu. That's because it's a way for us to go back in time and think about life and how it relates to the rest of the universe through that lens of biochemistry. Meteoroids and asteroids like Bennu are the closest thing we have to that primordial planet material. That's our best time machine back to the early Earth, is by looking at the materials that would have made up our planet. And so I can't divulge a lot. What you see on the right there is the sample from Bennu. Since I only got my sample a couple weeks ago, I don't have any results to share, and I can't divulge any results, but I will say that there will be a lot of announcements in March during the Lunar and Planetary Science Conference. That's when a lot of people are planning to unveil a lot of their great results that we have so far. But I can tell you, and you've probably seen it in the news, lots of carbon. We're going to learn a lot about the chemistry we share with the solar system through the many, many grams of material that were brought back by the OSIRIS-Rex mission. So this is probably the happiest picture there ever was of me. This is installing my volatiles collector that is on the bottom of the glove box that contains our pieces of the meteorite Bennu here on this planet, and is collecting data for me even as we speak. And I wanted to thank everyone who braved the weather to sit in front of your computer and join me on this trip through space and time. And I think there's a whole lot of stuff in the chat.
Yes, and you did such a great job. There are very few questions.
Oh no.
So really the only question is, how can we be sure that we know all of the elements?
Oh, that's a great question. Yeah, so this is part of a lot of experimentation that happens. You know, we know the number of subsets of things that go into elements, neutrons, electrons, protons, and we can build those up in a really predictable way. But then much like organisms themselves, bigger and bigger accumulations of those subsets that make up the elements become harder and harder to create in ways that are probabilistically unlikely outside of directed systems like organisms. So a lot of those things that you see on the periodic table that are rather large and have a lot of those subunits are actually manufactured, and they may be ephemeral. They may be made out in stars. They probably are being made out in stars, but they don't last that long. They don't even last that long when we create them inside particle colliders or anything like that. They're very ephemeral, just because there's a lot of forces inside those atoms that make them pretty unstable. But yeah, the extent of the periodic table isn't necessarily known. And that's a big part of chemistry, is trying to understand the forces inside atoms that limit what are the naturally occurring elements.
Excellent. Thank you. There's a question from Donna who wants to know, are there any other sample collecting missions coming up?
Yeah, so there's Mars sample return, which is a really important international activity between NASA, the European Space Agency, and other partners around. There's also that will bring back, you know, all these tubes, like, I don't know, I think they're like 6 or 8 inch tubes from the surface of Mars that have been drilled by a rover, and all of these different lithologies, all of these different types of rocks that can help us interpret a bunch of things about the surface, including possibly the presence of life. I always like to reiterate to people, though, that just like I'm really interested in understanding the abiotic potential of chemistry in space, it's also really important for us to understand the abiotic potential for planets and geologic systems, because that's something that's very hard to reconstruct even on our own planet, where there's such a huge overprint of life. So even if we don't find life on Mars, we're understanding how geology is directed on a planet without life. There's also a mission to go to the moon, Phobos, of Mars, and bring back pieces of Phobos as well. And of course, there will be more lunar collections with the Artemis missions too.
And I'm a geologist, so Darren has a great question that I love. And that is, if we found banded iron somewhere else without other data, would we assume a biologic origin?
Ooh, you know, that's a really interesting thing. And we associate banded iron formations with organisms because we know the huge effect that organisms had on the chemistry of the ocean that caused that big oxidation state, such that iron was precipitating at the bottom of the oceans in a very different way than it was before the ocean was oxygenated. I think that would be a strong biosignature, but you would want to do the due diligence of making sure that that wasn't recording some other mechanism of introducing oxidizing conditions into that ocean that you're looking at. Is there some sort of strange tectonic activity that would have led to that? When we think of tectonics on this planet, we associate it with CO2. But remember, a lot of that CO2 coming out of volcanoes is because organisms have done a lot of work putting a lot of carbon into the seas that then goes down in those subduction zones. So you'd want to retrace the geologic history of that planet to understand if there's other oxidizing sources. Was there some sort of sulfur chemistry in that sea, for example, that would have given you that same sort of evidence of shifting redox conditions in an ocean?
Excellent. This is a great one. Chris again wants to know, what would you find in Bennu sample that would make you jump for joy?
Oh man. You know, I think for me, I'm focused a lot on reduced carbon. And I think something that I'm really hoping that we can understand in our lab group, and that will make us collectively as the astrochemistry laboratory here at Goddard jump for joy, is if we could understand a little bit more about the mechanisms that drive the particular shapes of amino acid molecules. Amino acids have handedness to them. There's a diagram behind me on the wall that's showing this, but amino acids are what we call chiral. If you took a molecule like those structures that I showed you, they're not superimposable like your hands, but they are superimposable this way as mirror images. And so we have left-handed amino acids and right-handed amino acids. And organisms use all one shape of those molecules. But when we look in astromaterials, we find a bunch of, almost racemic, we call it, which is kind of half and half, except for certain amino acids, which it appear have a lot of the same sort of one-handed chemistry that we do. And we don't know if that's possibly the mechanism that's making that inside that rock isn't random. It's being directed by the minerals that are in that rock, the starting material of organics that was in that rock, the energy that that rock has absorbed during its alteration process. And part of the thing that we're really wondering is how much of our shape biochemistry, not just molecules, are we inheriting from the stars? How much of that one-handed shape represents some sort of excess that might be in certain astromaterials of a certain age that were part of our history on this planet?
Good. Matthew wants to know, do you plan future missions while you're still working on current missions?
Oh, yeah. No, there's constant missions being planned all the time. And that's because the time scale of these things is huge. OSIRIS-Rex, the mission, was first proposed in 1999. And when you propose a mission, you get a lot of input back from reviewers, and they point out all the things that you would need to fix in order for this to be successful. And then you go about trying to fix those things, trying to plug those scientific holes, trying to build new instrumentation that would suit your purpose and make it successful. And that's a review process that can go over the course of 5-10 years until you get it right. And then the resources are given to that team to actually start building all that stuff. So you work all the kinks out through about a decade-long process before you even get to building it to then launch it. And so, yeah, so I'm part of Mission Concepts right now, thinking about the outer solar system and what we can do out there, even though I have a bunch of OSIRIS-REx samples back in the lab calling to me.
So our last two questions are a little more hypothetical and fun. Is there a way to deconstruct what we have now to origins? And if origins are thought to come from bombardments, then are we the answer?
Oh, that's kind of a cool question. Yeah, so the way that we deconvolve history is by kind of unwinding these evolutionary clocks and looking at commonalities between all the organisms that we know arose on certain time periods around, the Earth through its history. And so that's how we get back to what we think those starting materials are. Now, the big problem with that is we're still starting at fairly complex microorganisms that have been in a constant conversation with their environment as a species for 3.5 billion years. And so they've accreted all of this new information. So it's very hard to understand what that original set of biochemistry might be. That said, even then, a lot of the fossils that we're looking at don't give us a lot of chemical information. That's not usually recorded in rocks because rocks get hot, they get pressurized, they move around. You lose a lot of those molecular structures that could tell you something interesting about biochemistry. So there's kind of a stop on how much chemistry can tell us. So we mostly do it by looking at organisms and where we think they are in the evolutionary time period. But yeah, I think I don't know if we're the answer, but I'm going to put out a challenge question back to that questioner, which is I would challenge the idea that there's only been one origin of life. There's no need for us to not think of the fact that there might have been several origins of life in the past or new life evolving right now, even in our own planet. We are overprinting the biology that we see here as a very successful biochemistry that we see all around us, that we share with organisms around us, but the same energy and elements and molecules that created life back then could be doing it right now in some unseen corner of our planet. And if we rely too much on analogy and what we see in organisms around us, we could miss interesting organisms even on our own planet or very close to our planet in places like Mars.
And that's almost a great segue to the last question from Kieran, which brings relativity into the conversation. And knowing that we're looking at light from distant planets and knowing that we're looking into the past, how does that affect our understanding of what our understanding of that life is?
Yeah, you know, I don't actually work on a lot of stuff outside the solar system. I'm just now joining a lab group that's the Virtual Planet Laboratory that's focused on exoplanets. And what I can say is, like, this is something that I think about all the time when they talk about their work. Like, oh my gosh, if we see these gases around this planet that close to its star, that's a very unlikely combination of gases. That would be a really strong biosignature. But that far away, is it a biosignature we're ever going to interact with? Could it have already gone extinct? Are we looking at fossil life in the forms of these atmospheric gases? That's what really kind of boggles my mind about exoplanet science is that's really just saying like, oh, it is possible, not like, oh, it is there and we're going to be able to visit it, because it could be on a, you know, in another time altogether by now.
Got time for one more, Heather?
Sure.
Okay. So the last question, can we terraform Mars?
Oh gosh. That's a really, I mean, I think that could we? I mean, we have a hard time keeping the potholes filled on this planet, right? I think something that is a little bit uh, overwhelming, but important to think about when we talk about terraforming is that terraforming isn't something people do. Terraforming isn't a human activity. This widely habitable, cozy planet that we live on is this huge accumulation of biology over 3.5 billion years that gave us this ocean chemistry that we're all adapted to, that created all of this huge reservoir of oxygen that we're all breathing, that gave us all of this dissolved organic carbon in our oceans that can be taken up by organisms to make biomass, to fill our oceans with our fellow organisms that can become part of our energy system, food. So we are this huge inheritance of biology, and it's far too easy to imagine ways of pushing around dirt that obscure the true job that biology has performed on this planet by making it the world it is today. We are in many ways very unaware of all of the activities of the 3.5 billion years of biology before us, because we're often very focused on just what we as humans have done to modify the planet to our liking, when there was billions of years of organisms that did a lot of hard work of taking this rocky planet. Remember when those Rhynie chert fossils showed up, those little 3 centimeter things, there was nothing on the surface of the continents. There was literally nothing. Think about what that meant for even maintaining soil, for having water and lakes. If there was no catchments, you know, it would have been a very different world. So sure, we could, but I think we're not understanding the scale at which it takes to actually modify a planet, because we barely understand the way that life has interacted with the planet that we're on right now. So that's back to time machines now.
Thank you so much, Heather. This was a fantastic talk. I really appreciate you weathering the storm and being with us, and I really appreciate everybody else who did the same. Thank you, and I hope we can have you again at some time.
Oh, absolutely. This has been a real pleasure. Thank you, everyone, for sticking around.
And hopefully you get home safe.
Yeah. Thanks all.
Thanks, Heather.
Thank you.
Good to see familiar faces too.
Presentation.